Bismillahirrahmanirrahim. Now I will discuss the overview of the process by which the cell-mediated immunity and the antibody-mediated immunity are induced by exposure to any antigen like a virus. You can see that over here a virus is present, and this virus has been phagocytosed by a macrophage. Now this macrophage will produce a cytokine that is called interleukin-1. When the virus enters the macrophage, it is degraded into small products, and one of that product, that is a processed viral antigen, is presented as an epitope. Now we must know two things: the major histocompatibility complex and the cluster of differentiations. This is MHC, that stands for the major histocompatibility complex, and CD stands for the cluster of differentiation. The major histocompatibility complexes are specific for the helper T cell and the cytotoxic T cell. The MHC1 and the cluster of differentiation 8, MHC1 and CD8, are specific for the cytotoxic T cells, and the MHC2 and the CD4 are specific for the helper T cells. When the macrophage phagocytoses the viral antigen, it then presents this viral antigen as an epitope in association with the MHC2 protein. This MHC2 then recognizes the T cell receptors that are present on the surface of the helper T cell, and it recognizes this helper T cell by the presence of this CD4 that is present on the surface of the helper T cell and helps in the recognition of the helper T cell. Once the macrophage has produced the interleukin-1 and it has processed the viral epitope in association with the major histocompatibility complex 2 type protein, it then binds on the T cell receptor on the helper T cell, and this helper T cell gets activated. When the helper T cell gets activated, it produces 4 functions. The first one is the B cell. It activates the B cell by producing 2 interleukins, that are the interleukin-4 and the interleukin-5. This causes the activation of the B cell, and it gets converted into a large plasma cell that releases antibodies. The second function is that it causes the activation of the cytotoxic T cell. This cytotoxic T cell has a receptor called interleukin-2 receptor. So when the helper T cell produces interleukin-2, it acts on the cytotoxic T cells, and that is recognized by its CD8. The major histocompatibility complex type 1 is associated with it. Once the cytotoxic T cell gets activated, it produces perforins that cause the death of the entire cell. The third one is the memory T cell, which gets activated by the interleukins, and the interleukin-2 also causes the further activation of the helper T cell. This is all. Thank you so much.